Hello, welcome to Introduction to Psychology. I'm Benjamin Miss, and we are talking about the nervous system. This is the biopsych, physiopsych part of IntroPsych. Our goal for this section is to understand really the building blocks of the brain and the brain itself. That's the neurons and the parts of the brain. So we want to be able to kind of sketch out and label neurons, and the same thing for the brain, and also understand how neurons work. Remember, this is important because somehow you take these bundles of cells, and here we are, your sense of self, your consciousness. And to understand this is to understand ourselves. Key things to consider here. Genes and inheritance. How do we put all these things together? We remember a lot of this from probably high school bio. We have 23 pairs of chromosomes. Our DNA makes up our DNA. RNA is copies of it that's used to create proteins. And genes can have either dominance, incomplete dominance, or codominance. For instance, we look at something like Huntington's disease, where a single copy of a gene can cause this disease. The cleft chin is a single gene. An eye color, in quotes, because there's a lot of variables involved, but oftentimes we consider it a single gene. Incomplete dominance is something like you can have red flowers, white flowers, and if you have one of each gene, you get a pink flower, which is basically like a light red, a combination of these two. And then codominance would be like blood groups, A and B. If you have one gene from a parent that's an A and one B, you have type AB blood. Both of them are this. The thing is that most complex traits involve many genes, and things like personality, this is really a complicated thing. Well, we go back to recalling Ramon y Cajal, who was the father of modern neuroscience, for his work with neurons, and we see here's an image of one of his drawings. This is from a cat. Right, an image from a cat. And so we're looking at how these neurons are kind of configured, what you see through his microscope. Well, we know that you have approximately 100 billion neurons, and that each neuron has about 7,000 connections to other neurons. These are synapses, which means 100 to 500 trillion. I know the numbers don't quite add up, but there's reasons for that. But the idea is about 100 to 500 trillion synapses, but a 3-year-old has approximately 1 quintillion, which is a very large number, far more than we do. And we wonder about why that is. And the reason has to do with the way that a developing brain works, right? A child has more synapses, but less organization. Now we look at the two main types of cells in the brain, which are neurons and glia. In this image in the middle, you have neurons, a couple different configurations. And a key here is that neurons do come in different types, different configurations. And they have configurations based on what they need to do, how many branches they have. And then in the upper part, we see tissue, brain tissue, which has, again, neurons and glia. You've got cell bodies, axons, dendrites, but it's quite messy. And one of the things we note is Cajal did an amazing job of being able to see these things and deduce these structures. Well, here we see another example of neuron configurations. And we see that our traditional view of the neuron is really quite different. If you look at these four, which one looks like our traditional view? And that would be the multipolar at the end. And so that's what we'll look at. Basically, at the top you have the cell body with the dendrites, and at the bottom you have the axon. The issue is when you look under a microscope, it is not always easy to see this. So we'll simplify it even more. Here's our neuron. And so what we care about here is, right, the top, you see the cell body with a nucleus, and then you see these branches coming off it, which are the dendrites. And the dendrites are what get information from other cells. So usually axons synapse with dendrites, form synapses with, And so you can imagine the cell forming 7,000 connections with other neurons and getting a lot of signals. Well, these synapses formed by connections with the axons from other neurons and forming connections itself with its axon, connecting with other dendrites. And so you have the two main things that I've mentioned here. One is the dendrites and one is the axon. And that's the key. Dendrites receive information and axons send information. And the neuron signals through itself electrically, and then it communicates between neurons. Here's another image of our different types of neurons that are really quite varied. So a very common type of cell is this pyramidal cell in the cerebral cortex that we see here. And then we see the Purkinje cells in the cerebellar cortex, in your cerebellum, where you have the majority of cells in your brain. The small structure at the back of your head contains the majority of the neurons in your brain by a lot, like 70 billion to 12 billion in the rest of your brain. And then the other one is the neurons in your olfactory bulb. So neurons have wildly different configurations, but what they all have are axons and dendrites, and the structures in there are similar. Now, when you look at that, this is going to give us an example of how these things kind of are set up. And there's a lot going on here. So let's unpackage this a little bit. So what do we need to know? If we're really going to study the brain and neurons, we need to understand how neurons signal. But we're going to keep it very simple. The reason we need to understand this is so that we can understand how neurons communicate. But it is quite detailed. You see here in the image, you've got K+, Na+, and Cl+. And there's something that's fairly counterintuitive for most people in regards to how neurons fire, and that's this. You have positive potassium ions inside the cell. You have positive sodium ions outside the cell. And when a neuron fires, it is these two that move. Sodium is going to come in and potassium is going to go out. So it's a movement of all positive ions. But what happens is this. You've got channels that are specific for for potassium and for sodium, specifically for these two, right? And so, at rest, you're more negative inside and more positive outside. So you open a sodium channel and sodium rushes in, making you very positive. And then after you fire, you open your potassium channels. And potassium now wants to rush out because you have way more positive inside than outside. And you also have this gradient. It's a very complicated combination of things and a very complicated balance. You are complicated because you have two gradients in place, concentration and charge, which leads to some of this difficulty. So this is kind of what we need to understand, is that firing along the axon is electrical. All right? This is a variance in the electrical, in the concentration of these charges that allows the signal to be sent. And that when the neuron is depolarizing and firing, you are having these sodium ions rush in because you have way more sodium outside than inside and way more positive outside than inside. So when you open that channel, sodium wants to go from high concentration to low concentration for both charge and sodium concentration. When you open your potassium channels after that, you've got way more positive inside and way more potassium inside, so the potassium wants to go out. So that's how the neuron is basically firing, which is really incredibly simplified and really ignoring a lot of information. So, but again, in order to kind of keep this, fairly on focus, because if we want to understand how this works, we want to understand how other things are affecting us. And so we'll use some examples. So let's go through the electrical and chemical communication and then look at how these things tie together. And I think that'll help us to unpackage and see how it works. Here's now the synapse. A is the axon and B is the dendrite. The little thing labeled 4 is the synapse, the space between it. And those tiny little circles in the synapse represent neurotransmitters. So when the signal reaches the very end of the axon, after the neuron fires, it causes these neurotransmitters to be released. These neurotransmitters then float they diffuse across the synapse, which is very tiny, and they then do something with the dendrite. And often this can be opening a channel to cause that neuron to then fire. Well, when we look at neurotransmitters, you're familiar with some of them. The most common neurotransmitter people mention is serotonin. And that should make sense, because when we think of neurotransmitters, we hear about this one a lot. People take serotonin drugs that affect serotonin for depression. But it's also involved not just in mood, but in sleep, aggression, and appetite. Dopamine is associated with mood, sleep, learning, and pleasure. So drugs that people take for recreational purposes will often affect dopamine. The two main neurotransmitters in the brain are GABA and glutamate. These are both the amino acids. So they're common, and they're old, and they have these very widespread effects. Beta endorphin is associated with pleasure and relaxation. One of the ways that we know about neurotransmitters is by drugs. So we will talk about how drugs affect the brain, because endorphin comes from the combination of the words endogenous and morphine. Endorphins are endogenous morphines. Endogenous means from within. So they're basically morphine-like chemicals that exist in our brains. We release endorphins when we're exercising, pleasure. They cause us to relax. Runner's high is a blast of endorphins. And then acetylcholine is associated with arousal in the brain and muscle movement. Your muscles, the synapses between your neurons, your nerves and the muscles are acetylcholine synapses. So these are really just a sample of neurotransmitters, but ones that we should probably know that are fairly common. So other behaviors that release dopamine include sexual excitement, gambling, video games. Really, when we think of addiction, we're thinking about dopamine. People with major depression show a less than normal response in the nucleus accumbens. So this is the little structure there in the basal forebrain. So when the brain is developing, it starts out as a tube, and it ends up kind of curving around, where like my fingers are the tip of the tube, and then they curve around like this. So now this is kind of what our brain looks like, with the lobes of the brain and the basal forebrain being in here. This is where the dopamine system runs through. And so we think about depression, we think about, well, serotonin drugs, but in reality, it's much more complicated than that. And there are so many other treatments and possibilities, because what's happening in the brain is beyond a simple neurotransmitter. Well, they also found that addictive substances that affect dopamine and other synapses, when you directly stimulate dopamine, you cause a massive pleasure response. So this little rat here in the box, you can see the image in the bottom here, is in the Skinner box and is pressing buttons. What they do is they train rats to press buttons through reinforcement. They look at schedules of reinforcement, and we'll see this when we go into conditioning. But this is very simple in this regard. They took an electrode and put it into the rat's brain around the nucleus accumbens, called the septal area. Then they hooked this electrode up to a lever in the rat's cage. So whenever the rat pressed the lever, the rat got basically a stimulation in that area of the brain, which caused a release of endorphins to the levels associated with 10. They estimate like 10 times the power of an orgasm. So this rat is then hitting this lever, and it's like, this feels great, and it keeps hitting the lever. Well, it releases these dopamine blasts. It's incredibly pleasurable, and the rat would keep pressing the lever until it passed out. They would then put a lever in the box that gave the rat food. The rat would ignore that lever completely. and keep pressing the dopamine lever until it died of starvation. Assumably happy, but still dead. When we think about drug use and addiction, it's not that far off, where people do engage in very similar behaviors. So how do drugs work? Well, now we're looking at a synapse again, but we've turned it sideways. So on the left of the image, we have the dendrite, and on the right, we have the axon. And we see there's a number of ways that we can get more neurotransmitters in there or affect neurotransmitters. So you want to boost dopamine. Well, certain psychoactive drugs may block a reuptake transporter. Caffeine affects dopamine systems by blocking the reuptake in a similar way to cocaine. But caffeine does it in a more mild way, where cocaine blocks that dopamine transporter in a big way. Okay? Nicotine, sends a signal to release dopamine. And addiction works on these kinds of signals. Now, when we look at this, we're talking about blocking the reuptake. So you release dopamine, and then dopamine is recycled and reused. But what happens if you block the reuptake? Well, this means there's more dopamine floating around in the synapse, and you're getting more of a dopamine effect. Interestingly, serotonin reuptake works the same way. Drugs that are given for depression, called SSRIs, are selective serotonin reuptake inhibitors. And what they do is they block the serotonin transporter, which we can imagine looks similar to the dopamine transporter here. So now we imagine this is a serotonin synapse. And so you have serotonin being released, and the drug that you take is blocking the reuptake. More serotonin is floating around. But over time, just like with addiction and drugs that we take for, people take for recreational purposes, the brain says, well, I don't need to produce as much dopamine, or I don't need to produce as much serotonin because there's more floating around in the synapse stimulating the axon or the dendrite of the next cell. So you've got this ongoing stimulation, and you don't need to produce as much. So we imagine that this could be problematic. We're producing less of something that we need. And so over time, the brain adapts. And with addiction, that's exactly what we see. The brain adapts. Methamphetamine causes the release of dopamine. So you're just releasing more. You can still bring it back in, but you're releasing more. Which, again, what's going to happen over time? Well, you're going to simply produce less dopamine. Part of the issue of addiction is anhedonia, feeling less pleasure. People don't feel as well because you take a drug, you feel good, then without the drug, you don't feel so good. So it's kind of like stealing from the future version of yourself. You're saying, I want this dopamine now, but then later, you're not going to have it. So you end up, for many people, into a cycle of addiction where they continue to take this substance. Drugs like heroin or nicotine may mimic the effect of neurotransmitters on the neuron. And so they look like the drug itself. In fact, nicotine affects the acetylcholine system and is called a nicotinic. There's basically receptors called nicotinic receptors because they found that nicotine stimulated them. And then they found that these were associated with acetylcholine. So we also understand that drugs can work in another way too. Drugs that people take for psychosis, right, like certain effects of schizophrenia, will block the receptors. And so people won't get the same effect. And this can also reduce the experience of pleasure. When we look at how these work, amphetamine, we said, releases dopamine, creates excitement, alertness, positive mood, and energy. Cocaine blocks dopamine reuptake, so you have more dopamine available in the synapse. Same effects as amphetamine. MDMA, this really works with both the stimulant and sensory distortion, commonly known as ecstasy. And this releases dopamine, serotonin, but it damages the axons. And so this is one of the most dangerous drugs that a person can take in a non-clinical setting, because under the conditions where it's taken generally, it amplifies the damage. So you end up with permanently damaged neurons. Not all drugs permanently damage neurons. You can actually create kind of like a hierarchy of how dangerous drugs are. Ecstasy, one of the worst. Bad for your brain. Heroin mimics endorphins. It leads to relaxation, pleasure, and euphoria. But it also means that over time, people are going to produce fewer endorphins and over time feel less pleasure without the drug itself. Heavily addictive, less damaging. Interesting how things work. Well, a couple ideas we get from how neurotransmitters work are the way that they affect the neuron. And when the neuron fires, you have the all or none law. So the neuron's getting these signals from all over, from its dendrites, and eventually it reaches a point where it fires. And once it fires, it either does or it doesn't, once you reach that threshold. And we'll look at what that threshold means. But the chemical communications between the neurons, along the neuron, it's electrical. It's those sodium and potassium ions. When neurons fire, the neurotransmitters bind to the postsynaptic terminal when you reach the end. And these can be excitatory or inhibitory. And you've got these two terms, depolarization and hyperpolarization. Well, it's not quite as simple as a neuron gets a signal and it fires. Because you've got sodium channels, potassium channels, you've got excitatory, you've got inhibitory. And so a neurotransmitter might say, hey, open up a channel. Open up a sodium channel, a bunch of sodium rushes in, depolarizes the neuron. But it also might say, block a sodium channel. Or open a potassium channel. Or it might do none of those. It might say, hey, we're getting a lot of stimulation here. Let's create some new channels. Let's attach to something that then eventually goes to the nucleus and activates a gene. So there's a lot of different things that can happen with neurotransmitters. But let's just look at the way it fires. So one of the things to note is firing rate matters. Neurons have a natural base rate of firing. You take a neuron and it might fire 1 time per second on its own. As you dump some neurotransmitters into that synapse, it might increase the rate of firing. So now it's firing 10 times a second. Or you increase it even more, now it's firing 20 times a second. What's interesting is the neuron can have a different effect based on how fast it's firing. What we see on the other side of the image, down at the bottom, is the electrical potential as it fires. If it's a resting state, it gets stimulated, and when you reach threshold, it then depolarizes. So you can have failed initiations. You can have it depolarizes a little, but it doesn't reach threshold. This is the all or none aspect. Once you reach threshold, boom, you fire. But until you reach threshold, you do not fire. So you need to depolarize. Let in some sodium, for instance, to depolarize a little bit. And then you reach the right amount and boom, all the way. Once you go to the top, you open your potassium channels. Potassium rushes out, taking that positive charge with it, repolarizing. Then you reach the refractory period where it's very difficult to fire again, and return to the resting state. Now, this is far more complicated than I've described. But this is the general idea we need to have just to understand what's happening with neurons and how complicated they really are. Sodium and potassium are positive ions, and the flow of them is happening while the neuron is firing. Firing rate is how fast the neuron fires, and these incoming informational signals are influencing that. Now we look at the delineations of the nervous system. So, the nervous system is all of our neurons. You've got the central and the peripheral. The central nervous system is the brain and spinal cord. That's it. The peripheral nervous system is everything else. So the peripheral nervous system is our spinal nerves and our cranial nerves. These are nerves that connect from our face and neck area generally, but directly to our brain, but are not part of the brain or spinal cord. These are involved in the autonomic and somatic nervous systems. There's a couple things here that are interesting, and the models are changing a little over time. The visceral nervous system refers to the things that control specifically your internal organs. And the autonomic nervous system and somatic nervous system are really considered the key parts here, where autonomic sounds like automatic. And that's a good way to think about it. Basically, this delineates more. The somatic nervous system is your muscle movements, your spinal, kind of muscles and nerves where you're kind of actually initiating movements. It's your conscious control. The autonomic is your fight or flight, for instance. And the two main delineations here are sympathetic and parasympathetic, where your autonomic nervous system, I said, think automatic. Sympathetic is arousal, and parasympathetic is relaxation. So fight or flight would be like your sympathetic nervous system. Once you're relaxed, your parasympathetic nervous system is going to, take over. And your enteric nervous system is often associated with the parasympathetic, but it's been described as something different because of its very interesting effects. But this is specific to your digestive system and your gut. And there's this new area of research that's looking at kind of like the brain in your gut, where your stomach has a lot of very interesting effects, and there's this like kind of feedback to your brain from this part of the nervous system. We can look at it here and see kind of the same things, but broken down a little more simply, where you've got two main types of directions, where you've got afferent and efferent, which are your sensory and motor. And we think of it as afferent sensory. You need to sense something before you can act, so A before E. Afferent, efferent. Sensing it and then moving. You've got your somatic nervous system, which is voluntary, your skeletal muscles. And then you've got your autonomic nervous system, which is your involuntary, which is your fight or flight, sympathetic, and parasympathetic. So think of parasympathetic like parachute, something to slow you down for that resting. Then we consider our spinal cord. And you've got these same things, the afferent and efferent neurons, but you've also got these interneurons. Think about reflexes. You would go see your pediatrician and they'd tap you on the knee with a hammer and you'd have that little muscle twitch. They were looking to basically see how well is that muscle functioning. Is it basically, are you getting a very fast signal? And they didn't care whether you kicked your leg or not. They cared if they saw that little muscle twitch because it meant that you had a reflex. Well, this reflex is happening in your spine. If you've ever pulled your hand away from a hot stove, this reflex is happening before your brain is aware that you are getting burned. This is something that's happening in your spine. Because in your spine you have, the same kind of tissue as you have in your brain. You've got these neurons. And the interesting thing is the inside of your brain is the white matter mostly, these connections, and the outside is gray. In your spinal cord it's reversed. Your nerves are connecting in the white matter on the outside, but inside you've got really these interneurons that are making these kinds of decisions. And you've got these tracks that run up and down to your brain and back. Well, we're kind of in a golden age of neuroscience where there's so much research that has been happening and things that are being discovered. And it's really quite fascinating. And up until the mid-19th century, this is the 1800s, people really didn't know that much about the brain. And there was a very famous case of Phineas Gage. Phineas Gage was working on the railroad in Burlington, Vermont in 1948. And this tamping rod blasted through his brain. It was knocking down into the ground, tamping down into the ground basically these explosives, and these explosives shot this rod through his brain, wiping out a great portion of his frontal lobe, taking out his left eye, and he had these massive personality changes. And it's been used as an example of understanding the brain. What's been interesting is more recently, research has been coming out that Gage was able to function. The story is he toured with basically a freak show with the rod showing what happened to him. And he ended up going down to South America and working as a coach driver. And he was able to function. And he was able to live on his own and do okay. So there's a lot that we still are understanding and still figuring out about this. But the myth of Phineas Gage is that he was no longer able to function at all. But what happened is around this time, we also got information from Broca and Wernicke and other things that told us what was happening in the brain. Now, we use locative terms about the brain. Dorsal means back. This is our spinal column and also the top of our brain, because for humans, our spine and our skull are kind of working differently. If you have like a dog, you've got like their back, and then their head is kind of like right there, right, along that same alignment, where that part of their brain is in line with their back. For us, because of our upright posture, it kind of curves. So the top of our head is also dorsal, just like our spine is. This is like remember like the dolphin, like the dorsal fin, the fin on the back. Our belly down here, and also down here, is our ventral part. And so you consider there's a couple things, like information goes to our back of our brain and then actually splits dorsal and ventral. These are called the dorsal and ventral stream of information. So we have to understand these terms. Lateral means toward the side, right, left and right. Medial means toward the center. And anterior is toward our head, and posterior is toward the tail, or, toward the back. So we describe parts of the brain using these terms. We also look at the brain through different slices. And so we've got different planes. You've got the frontal plane, which is also called the coronal. And I like the term coronal rather than frontal because there's a good kind of mnemonic to work with this. If you were to imagine putting on a crown and then coming straight down, that's the coronal cut. Well, here is we see the frontal cut. The sagittal plane is cutting through the center and looking at the middle. We look at that image a lot because it allows us to see structures in the middle of the brain. And then the transverse or horizontal with the ground, is another way of looking at this. So we want to look at what are the main components and what are the things that we need to understand. When it comes to brain anatomy, here's the main lobes and systems. So the four main lobes are frontal, parietal, temporal, and occipital. The frontal lobe is associated with decision-making, movement, and also you see this term inhibition. The frontal lobe is the part of your brain that says, that's not a good idea, don't do that. That's what we mean by inhibition, inhibiting our impulses, our limbic impulses. The parietal lobe is associated with sensory information and navigation. The temporal lobe is associated with sound, language, and processing faces. The occipital lobe, sight, vision. That's it. It's such a dominant sense for us that we even are using, our ventral and dorsal stream through our parietal and temporal lobes to do visual processing. And then you've got your limbic system, sometimes called the limbic lobe, but not really. This is emotion and memory, our impulses. And then we have the insula, which tells us about body states and pain. Well, hemisphere, right? We have two halves of the brain. These are called hemispheres. And we've got a gyrus, which is a raised area, and a sulcus, which is a depression or a fissure. Gyri and sulci, the folds in the brain, account for the large proportion of the neocortex. So these folds are important, these gyri and sulci, because if you had a perfectly smooth brain, you would not be able to function at all. This is the higher level processing that's happening on the surface, and these folds create more surface area. In the same way, harbors, right, give you more coastline than simply a straight coast. A fissure is a large sulcus, and the space here between the frontal and temporal lobe is called the sylvian fissure. It's a very deep sulcus that delineates between those structures, and if you were to pull that back, you would actually see the insula inside, under there. The ventricles are spaces in the brain where CSF, or cerebral spinal fluid, is produced. Cerebral spinal fluid is like what's in your, not just in your brain, but also in your spinal column, where you have this fluid that helps to kind of protect and also allows you to get rid of things. Like it eventually is going to merge into your blood and dump into your blood toxins and stuff that build up in your brain over time. When they do a spinal tap, they're going into your cerebral spinal fluid to take some of it to see how it is. We've got your, if we go deep into your spinal, really the top of your spinal cord gives us your brain stem. And you've got your cerebellum, your pons, and your medulla. Cerebellum, I mentioned, it has the most neurons in the brain. It's involved in coordination and precision of our movements, and more recently discovered to be shown involved in even things like memory, heavily involved, attention, all sorts of aspects. Pons is Latin for bridge, and it links the medulla and the cerebellum with your thalamus. Your thalamus is like the center switchboard of the brain, which we'll see. The medulla is doing a lot of those automatic functions that we kind of take for granted. Beating your heart, breathing. In your midbrain, it's a very small area. I know we say midbrain, but midbrain's an old structure, and in some earlier species, if you look at a fish brain, their proportion of midbrain, hindbrain, and forebrain is fairly even. But for us, the midbrain is about doing what it did historically. It's your forebrain, your cerebral cortex, that does a lot of your kind of like newer, higher level processing. So your reticular formation is sending signals about arousal to the rest of your brain. And you've got in the midbrain what you call the superior and inferior colliculus. The superior colliculus is automatic attention, and the inferior colliculus is automatic auditory perception. So if you were to see something like out of the corner of your eye, like poof, and you look toward it, it draws your attention before you're consciously aware of it. This is a deep midbrain function. The thalamus, I mentioned, is right in the middle of the brain, not the midbrain. It's part of the limbic system. It's involved in the relay of information, arousal, alertness, and possibly even responsible for consciousness. The hypothalamus is below the thalamus and involved in what's called homeostasis, which are things like maintaining your blood pressure, your body temperature, using a lot of your resources to maintain you in a fairly narrow window of stability. It also is in charge of producing stress, growth, and sex hormones, or telling your adrenal glands to produce those things. The amygdala, associated with emotion and your impulses. And the basal, and also right next to your hippocampus, which is involved in memory. So it's sending a pretty powerful signal about emotional ties to memory. Basal ganglia are deep and involved in motor control. They kind of wrap around the thalamus and are involved in the dopamine and learning systems. Specifically voluntary control and learning, a whole memory system about procedural learning. They're also involved in Parkinson's and Huntington's disease, major movement disorders. The cingulate cortex is above the limbic system, between the limbic system and the rest of the cortex. This is involved in, it would make sense, early processing of emotion and aggressive behavior, because it's near these impulse centers. The septal area nucleus accumbens we saw is involved in pleasure and reinforcement learning. And our hippocampus involved in memory, specifically autobiographical memory, and related to memory for facts and information. Now we're looking back at the surface of the brain, and we see Broca and Wernicke's areas. Broca's toward the front of the brain, anterior, and Wernicke's toward the back, posterior. Broca's area is involved in producing language, and Wernicke's in understanding. When we consider what's happening in the brain, we consider how vision and language are heavily involved and tied into a lot of systems. And then we consider that it's possible for somebody to even lose a large portion of their brain and still function. However, it's better for younger to have that damage in terms of recovery. Some early work on the brain showed that early brain injury including removing large portions of the brain, people can recover much better the younger they are. Now let's see how we're doing. If we look at the brain, we say, okay, here is the lobes and major structures. Can you look at this brain and identify frontal? Well, the frontal is on the left of the image, right? The anterior part of the brain, pretty large. Parietal is just behind that. There's a sulcus here kind of delineating between frontal and parietal. So parietal is around here. And then temporal is this piece down here at the inferior toward the ventral surface. And then in the the posterior, you have the occipital lobe. Below that, at the base of the brain, you can see the cerebellum, and here a little bit of the brain stem. Their motor cortex is at the posterior portion of the frontal lobe, right in front of the sensory cortex in the parietal lobe. And the auditory cortex is at the very top of the temporal lobe, right in the middle of this top part of the temporal lobe here. Now we look at the neuron. And there's a few more terms here than we discussed earlier. So you see on the left, you've got your dendrite, and on the right, you've got your axon. And also we call axon terminals. These are what make connections with your other dendrites, right? Form synapses. We see the cell body in the nucleus, the dendrites in the axon. We see what axon terminals, and then there's a couple more terms here. Myelinating cells and node of Ranvier. Well, many neurons are insulated with myelin. Myelin helps them to fire faster because the signal now doesn't have to travel all the way down. They don't have to open up channels all the way down. They can jump to channels along the nodes of Ranvier. So these yellowy pieces represent cells that are wrapping around the neuron, and then you've got these little nodes between these that are where it's jumping to. How we know things is through neuroimaging. CT is like an X-ray of the brain, and what you have is basically slices. It's also known as a CAT scan. So you can see different areas of the brain in pretty good resolution. PET is positron emission tomography, and what this is doing is using a radioactive substance to see where things are active in your brain. MRI is magnetic resonance imaging, which gives you a pretty good image of the brain, but this is only using magnetic fields to cause some of your atoms to line up. It looks at the density of tissue in terms of where there's water, and so you can get an idea of tissue density. CT and MRI are used to look at different things. Some may be stroke or bleeding in the brain, depending on the type of test. fMRI is functional magnetic resonance imaging. And by functional, this is a newer thing. It's been around since the 1990s, where you can look at where in the brain you're active. A lot of new research is coming from fMRI, but even newer things are available now. EEG is electroencephalogram. It's an old method of neuroscience. These are the ones that they put electrodes or an electrode cap on your head and measure what's happening. They can't see where in the brain things are active, but they can see that there is activity, and they can see change. So EEG can give you very rapid information on change. fMRI can tell you approximately where something is happening. And those two together give us a lot of current information about the brain. Our next piece here is the endocrine system. Hormones. So our hypothalamus is basically controlling this. You see here the pineal gland and the pituitary gland. Well, the pituitary is connected to the hypothalamus, can also almost be considered part of it as part of the brain. And you've got a direct connection that's causing you to release hormones. And these release hormones into the blood that then reach these other areas that release hormones. And so your pituitary is releasing, basically kind of commanding the rest of these and communicating with them through hormones. The thyroid is releasing growth, metabolism, and appetite-related stuff. The adrenals are situated over the kidneys, involved in stress response, adrenaline and noradrenaline, which are neurotransmitters. The pancreas is regulating blood sugar, and the gonads are regulating our sex hormones. So this should give us a pretty good idea of what's happening in our brains and how neurons work. This has been very brief, and there is a lot of information here. So I hope that you have enjoyed this part of our discussion and reviewed this information to make sure you understand what it is that makes us us, and that's our brains made up of neurons. Thank you, and I look forward to our future discussions.